biology. Ooh. So the context is a serious one, which is that a lot of my work is on conservation policy, the history of conservation policy and looking forwards in the way it works. And there is a major problem globally of biodiversity loss. Biodiversity interpreted usually under the definition of the conventional biological diversity from 1993 as the variability among living organisms from all sources, including terrestrial, marine and other aquatic ecosystems, and the ecological complexes of which they are part. So diversity within species, between species, and of ecosystems. And the conventional biological diversity has been around now since the 1990s. The 15th meeting is going to happen of the parties to the convention, going to happen in China in October. It should have happened last year, and it's been delayed for a year. And at that meeting, the primary thing that will be discussed is how the conservation of biodiversity should be organized. And the thing I want you to understand is that biodiversity is now understood as a global problem. Conservation action is global. And we've seen astonishing investment in the protection of biodiversity. So the area protected within nature reserves or national parks doubled in the 1970s, the 1980s, and the 1990s, and it's gone on increasing since. There are now about 16% of the Earth's land surface within protected areas. In 2010, the convention set a target at its meeting in Aichi in northern Japan to have 17% of the terrestrial and inland waters protected by 2020 and 10% of coastal and marine. They didn't quite get there, but it's been a remarkable process. And at the meeting in China later this year, they're going to set targets, even stiffer targets for the period post-2020. So international effort focused, biodiversity surely saved. Well, not quite. Biodiversity loss is unchecked. There are lots of measures of this. The one that works is the Living Planet report from the Worldwide Fund for Nature, WWF. And this is an index that they've done of the abundance of just nearly 21,000 different populations across a range of taxa from 1970 onwards. And I mean, the obvious thing about it is it's declining, and it's declining continuously year on year. So more and more money is being spent on biodiversity protection, and yet biodiversity loss is accelerating. And the reasons for that are many. This is what the Living Planet report identifies as the key questions. Changes in land use, basically the loss of habitat, particularly tropical habitats, overexploitation of species, particularly in the marine zone, but not exclusively, invasive species and disease, pollution, and climate change. So a whole host of different kind of factors. And you can see that there are different threats in different parts of the world. And a lot of the conversation in conservation is what can be done apart from setting targets at international meetings, what in practice can be done to lessen human impacts on Earth, because almost all of these impacts, these drivers of biodiversity loss are driven by human society, the world economy, the wants and needs that we all express through our purchases, through the things we eat, the things we buy, the things we do. And the sort of way in which they're talking about it, this is a nice paper that was published in last autumn called Bending the Curve. How do you bend the curve, the terrestrial biodiversity loss, from its sort of secular decline into some other kind of trajectory? If you do nothing, it will go on declining. The curve will slowly flatten because there'll be less species to lose. If you spend more money on conservation, you can do something, but you can't, you're still going to suffer a lot of loss before you get a certain recovery. Or else you can actually sort of try to influence the patterns of production and consumption. That would lift it further. The diagram is completely schematic. There are data, but there's no data behind this diagram. So biodiversity loss and the way in which conservationists respond to it through increasingly shrill demands for more expenditure and starts their critiques of the way society works are really quite important. Why synthetic biology? Well, simply because the scope of applications is huge, as you will probably all know, and as scientists and engineers sort of lift their eyes from the micro scale of the technologies of genetic engineering to look at different applications, the applications seem to replicate. So Venki Ramakrishnan of the Royal Society, speaking back in 2017, talked about a new age of biology based on genetic technologies. And he outlined a whole series of areas: human medicine, obviously, also all sorts of work on genes to genetically based diseases, all sorts of applications in public health, particularly insect vector control, but many other things. Lots of applications in agriculture to increase productivity, to control diseases, to enable farm animals to survive under the kind of industrial conditions that the world economy prescribes that they should be brought up in. Biofuels, biopharma, and last and perhaps rather small on this horizon, biodiversity conservation. So there are conservationists who started saying conservation needs a bigger toolbox. It can't work with its conventional methods of protecting species and protecting the spaces where they live. It has to do something else. And along with a friend of mine, Kent Redford, a biologist, a conservation biologist from the US, back in 2013, we had held a meeting in Cambridge, looking at synthetic biology and conservation. How will synthetic biology and conservation, how might they interact to shape the future of nature? And since then, we've been working on, and we've just produced this book, which we called Strange Natures, Conservation in the Era of Synthetic Biology. And the question we really ask is this: is synthetic biology simply another tool for conservationists to use, or is it in some way different? And the reason we ask that is that quite clearly there's a strong history of public disquiet about biological engineering below the level of the species, with the rather awkward history of GM crops being very fresh in most senior scientists' mind. Is it another tool, or is it something different? If it is a tool, where and how should it be used? What will be the consequences of its use, and who should make those decisions? That's what we look at in the book, and I'm going to say a little bit about how that might work today. Well, first of all, what applications are being applied? How would conservationists, how are they interested in the possibilities of applying novel genetic technologies to their conservation work? Well, the first one is in the control of invasive species. Invasive species are a major threat to biodiversity, but particularly in certain circumstances, like on oceanic islands, where you have a very highly specialized fauna and flora, mice and rats. So the unfortunate albatross chick, top right, on Gough Island, is being predated, is being eaten actually alive by a house mouse, and a conventional house mouse that you might find in a badly managed laboratory in a British university. And the house mouse was introduced some centuries ago from sailing ships, and it's now a major predator. And mice and rats are predators on many islands. So the island of South Georgia, right down here in the South Atlantic, had populations of brown rats introduced by the whalers who went down there or sealers and then whalers who went down there in the 19th and 20th centuries. And there's been a major program to eradicate those animals. And it's been done. It cost $11 million, about $100 per hectare. And it was done by spreading poison baits very densely. And there are proposals to, there's work of that kind in many parts of the world, perhaps particularly in New Zealand and Australia, where the indigenous fauna has been very severely hit by invasive species, many of them introduced misguidedly by European colonial settlers. And the New Zealand government has set a goal of a predator-free country by 2050, all those predators being introduced species, all of them being amazingly destructive. So invasive species are recognized as one of the big threats globally to biodiversity, and they're really expensive to do something about. So could you apply new genetic technologies to it? Well, yes, you could. Here's a paper that came out earlier in the year from the Roslin team led by the scientists at the Roslin Institute in Edinburgh, looking at the possibility of CRISPR-based gene drives. Where am I going? Sorry. To get rid of a species which is a real problem in the UK, which is the grey squirrel. Very cute, but very destructive. It came in from the USA. It was introduced deliberately on the grounds that it looked cute in those days, 1890. It expanded its range from southern England where it was introduced, and there are now 2.5 million grey squirrels in the UK. You can see its map spreading here from 1940 at the end of the Second World War, 1945, 2000 to 2010. It's spreading northwards. And as it spreads, the indigenous red squirrel, the native red squirrel, is disappearing. It also causes a lot of damage in forestry plantations. So cute though it is, it's a destructive animal. And in conservation terms, it's really tricky. There is competition between reds and grey squirrels, which basically turns around resistance to squirrel pox, which is a disease that came in with the grey squirrels and to which red squirrels are vulnerable. So grey squirrels move into a woodland, the red squirrels get sick and die away. And there are now less than 200,000 red squirrels in the UK, and they have a huge following, not least because of the place they have in childhood literature. Beatrix Potter's book, The Tale of Squirrel Nutkin, makes the cute little red squirrel the great poster child of British conservation. So how do you control grey squirrels? Well, you shoot them, you trap them, you poison them. Methods that Faber et al. Talk about as inhumane, labor-intensive, expensive, and ineffective. Really, really difficult. We have volunteers like this lady, an article in The Guardian, who shoots grey squirrels, but none of it's very neat. What Faber do is explore the possibility of a CRISPR-based gene drive. And basically a gene drive, as you know, in this instance, can change the chance of parental genes being inherited. You can attach a genetic element to that gene, and the traits are driven through the population. Gene drives occur naturally, but you can create them artificially. And in grey squirrels, you can do it so it cuts a site in the coding sequence of the female fertility gene. It's repaired but disrupted, so female offspring are infertile, and therefore the population declines. Would it work? Well, the paper suggests that it would. This is a modeling with different assumptions about mating, different assumptions about starting population size. But it would drive the population of grey squirrels, would be capable of driving it extinct with between sort of 12 years and 35 years. Of course, you know, if you have a lot of the country covered in grey squirrels, you get a lot of reinvasion, but it would certainly get rid of a lot of squirrels without covering the countryside with poison or bullets. So that's the kind of technique that catches the eye of conservationists. And they say, well, maybe you could do this on an island. Maybe you could get rid of invasive rats or mice without having to spend millions of pounds on helicopter time dropping pellets of poison in every square meter of the island. Maybe you could just do it by releasing animals in a sort of a neat, clever kind of way. So there's a whole series of applications which are basically like that, and many of them using gene drives to drive a particular population locally extinct. Secondly, people talk about using genetic technologies to attack wildlife disease. So there's something called chytridiomycosis, which is killing amphibians, particularly frogs, right across the world. And at the moment, there's no solution to it. And people are saying, well, maybe you could use gene editing techniques to alter the bacterial population on the frogs, on the amphibian skin, which seems to be what is really important in giving them resistance to infection. So you could find strains of frogs that seem to be doing rather better than others, and you could work out, you could sequence their genome, you could work out what aspects you need to fix. Maybe there's a technique here that, just like the disease is moving sort of seamlessly through the population, you could actually do some, and I've heard people use the word neat gene editing, and then you'd sort of repair the population. So wildlife disease is a big thing that people talk about. There's a number of, oh dear, sorry, my trigger finger has got static on it, I think. They talk about it for white nose syndrome in bats, which is another fungal disease which infects hibernating bats in caves in North America, and it doesn't kill them directly, but it weakens them and they die. And again, people saying, well, maybe you could fix that, give the bats a resistance to it. And there's also some work being done on other species like black-footed ferrets and sylvan plague. So if you've got either a very rapidly advancing disease problem, and wildlife disease is a major challenge for biodiversity conservation, a very rapidly advancing problem, or a very rare species, the black-footed ferret, was thought extinct until a very small population was discovered in Montana. In those circumstances, there are various sorts of techniques which are really thought of, are being discussed, actively discussed, and some actively researched. So that sort of the notion, there's quite a few places where people are proposing this kind of work. Another context which is very interesting is avian malaria. In Hawaii, as you probably know, it's a set of islands right in the middle of the Pacific. It has a very high level of endemism, and many of the endemic birds are already extinct, and the ones that are not extinct are threatened with extinction. And one of the main causes of death is avian malaria carried by mosquitoes. Both the malaria and the mosquitoes were introduced into Hawaii at some time in the late 18th or 19th century by European sailors. Conventional control methods are really problematic. Trying to use insecticides on the steep mountainsides where the remaining habitat is really difficult. So they're talking about other techniques such as the sterile insect technique, or again, engineering a gene drive to reduce or eradicate mosquito populations by biasing the sex ratio, or modifying the mosquito genomes, they no longer carry the pathogen. And there was a big meeting back in 2016 around the Society for Conservation Biology meeting, which was in Hawaii, looking at these options, looking at their scientific feasibility, looking at their social acceptability. So there's a lot of interest in particularly the potential for gene editing in these kinds of contexts. What else can they do? Well, the third thing that's being looked at, I call promoting adaptation to the Anthropocene. If you accept that we live in the era of humankind, the era when, during which Earth systems are driven by social and economic systems, human social economic systems, is there a role for gene editing of wild species? And again, scientists are saying, well, yes, there's a whole series of things we could think of doing. There is, for example, interest in engineering changes in the genomes of corals. Some corals, so you have a problem at the moment with climate change, that the oceans are getting more acidic and warmer. So they're thinking, well, that there are places where corals survive in warm water. Around Hawaii, for example, you get corals around thermal vents, which are living in much warmer temperatures, and temperature that would cause coral to die elsewhere. Well, if you could sequence the genome of the coral, if you could work out which parts of that genome give it that ability to survive, then maybe people are saying maybe you could put those genes or those elements into the gene, those sequences, you could engineer them in the genes of corals that live elsewhere, for example, on the Australian Great Barrier Reef. And so you kind of use your genetic engineering to kind of reseed, if you like, the whole of an ecosystem with species that are more resistant, that are virtually identical, but have this engineered component. And there's lots of other proposals here around addressing the larger scale demands of human society on natural ecosystems. In the field of energy, for example, biofuels from synthetic algae, or using gene editing methods to create synthetic meats. There's lots of ways of making synthetic meat, but some of them involve synthetic biology. Or, the creation of organisms to detect or break down pollutants. And those of you who have been watching the kind of progress of iGEM over the years will see these kind of ideas being experimented with by sort of free-thinking student groups. There are also questions about those novel biologies. I like this set of work. It's by a sort of biologist artist called Daisy Ginsberg, and she talks about the moment we're in between past natural biodiversity and future synthetic biodiversity. And she says we're in this crossover, and we should enter that period very, very thoughtfully. So she talks about, I mean, at the level of artistic design, she talks about the possibility of creating novel species which will work to undo the damage that human management of ecosystems do. So she has here something that looks rather like a sort of sea slug moving through a forest floor, and actually it's processing polluting materials and sort of cleaning the environment, and at the same time it sort of has a series of sort of sensors on its top that tells you what it's doing. I mean, it's a wonderful fancy that she offers. It's not a piece of genetic engineering, but nonetheless it's a sort of it's a conceptual frame for the thinking that people are starting to do. Here is her rewilded forest with completely novel organisms sitting in what is otherwise a fairly conventional piece of western oak forest from the UK. And there are people who push saying, well, actually, you know, it's not unreasonable to imagine the synthesis of whole ecological communities, microbial communities with desirable properties, where you have, you know, huge areas of cities with a substrate of concrete and all sorts of pollutants going into the water. We need to reimagine what kind of ecological community we need to remediate that environment. And those are the sorts of things. It goes well beyond the protection of existing species, but nonetheless, thinking is part of it. Or a nice paper on Earth terraformation, large-scale interventions in global environmental systems. The sort of thinking you'll be familiar with geoengineering. How do we respond to climate? Well, maybe we should, you know, change the reflectivity of the upper atmosphere, or maybe we should seed the oceans with iron filings to increase the amount of microorganisms. Similarly, people are thinking, well, synthetic biology could be used for these things. We could make these organisms, and those sort of ideas are there. And fourth and last, the one that catches all the headlines, which is what if you could bring species back from the dead, de-extinction. And there's been bits of work on this, lots of work. It picked up in 2013, it got the cover of National Geographic, which means that every journalist knows about the idea of bringing back extinct species. And of course, we all watched Jurassic Park, so you know, we all know about that sort of story. But there are serious efforts going on. There is some experimentation with trying to edit the genome of the Asian elephant to resemble that of the woolly mammoth. Beth Shapiro's book is quite good on what you can and can't do in the way of de-extincting a mammoth. But it's not impossible. Within a few decades, we'll have a very hairy, long-tusked, cold-tolerant Indian elephant on offer. And Revive and Restore, the US organization, has a number of other projects going on. One, for example, with the passenger pigeon to try and you know, convert, if you like, to convert a living bird back into the shape and form of something previous. And the point about all of this, and particularly de-extinction, is that it is, in conservation terms, profoundly weird. This is not what conservation has been doing, but the technology makes it possible. So conservationists have sort of walked up to a door marked sort of, you know, gene editing, and they've opened it. And inside there's all these ideas. What are they going to, and how should they think about it? What actually should be adopted as a sensible, practical, foolproof sort of strategy? And who should they ask? Those are the kind of questions that are of interest. So we talk about some of these things in our book. We also talk a bit about the science. The book is aimed in large part at people who work in conservation, and we don't reckon that they knew much about genetics. And indeed, it was a pretty steep learning curve for both Kent and I. And then we spent quite a bit of time towards the end of the book talking about how conservationists should respond to synthetic biology. And the first thing to say is there are an awful lot of particularly non-governmental organizations concerned in one way or another with these kinds of questions. And I just put this up. It's a social network diagram from work done at the University of Loughborough. And the environment is down here, but not all of these organizations are out and out conservation organizations. Some are very broad. So the Worldwide Fund for Nature, for example, is very much centered on biodiversity conservation. Greenpeace International is not. It's as likely to be campaigning about nuclear power or some other pollution, some other environmental problem. So, and there are areas within other aspects of global social movements that are interested in the environment. This hasn't got poverty on it, but if you had poverty, the poverty and environment NGOs interact around questions like gene editing very strongly. So the purpose of putting this up is not to get talking about social network analysis or the nature of the NGO world, but just to say conservation is not one single thing. And the opposition, which is around to the applications of synthetic biology in conservation, comes partly from the core of wildlife and species and biodiversity protection, and partly from other parts of the environmental movement. So the first thing to say is that it's a very mixed, very diverse playing field, and different organizations, different people will approach this in very different sorts of ways. It has been discussed. The discussion has grown since our meeting in 2013 in Cambridge, which was Kent Redford's vision, and I think really quite prescient. The CBD, the Convention on Biological Diversity, has been discussing it since 2014-15. They're still discussing it. They can't at the moment decide whether they should, or under what heading they should be discussing it. Should they be talking about it as in terms of a novel technology? Should it be talking about it in terms of rights to intrinsic variety of nature? So every time the CBD tries to talk about it and produces a report about it, it creates a flurry of controversy, and different countries take very different positions on this. There are countries that are very bold, very ambitious, very visionary, and countries that are very cautious. Broadly, the US and China probably are best known of countries that are really quite wanting to move forward very fast. The EU, while it has lots of outstanding science, the EU is much more cautious. The UK has gone from being important in developing the EU's position to now moving very strongly towards a US-focused position. There's also been discussion in IUCN, the International Union for the Conservation of Nature. They did a scientific assessment back in 2018. They also have a delayed Conservation Congress this autumn, and one of the items on the agenda is an IUCN policy on synthetic biology. And Kent Redford, my co-author, who's been very involved in that, says it's by no means clear whether they will get any kind of agreement as to what ought to be done. All of which is to say that there are some very exciting possibilities, but there's also a lot of quite challenging controversy around these things. The benefits I've outlined, and basically it comes down to an argument, the two arguments as to whether genetic techniques offer a fantastic toolkit or a Trojan horse. And these are the two sort of quotes that sit it. The critique, for example, of the release of genetically engineered American chestnuts, described by parts of the environmental movement as a massive and irreversible experiment. So it's pretty hard, it's already pretty hard to get either the biodiversity conservationists themselves or the wider environmental movement to look at these issues in a rounded way. And there's quite a bit of controversy already around it. It's clear that, and I think everybody agrees on this, that there are risks to the release of genetically engineered organisms into the unbounded wild, and that it's not a small thing to take a wild species and start to deliberately shift its genome. It's a form of domestication, if you like. So Venki Ramakrishnan comments, if advances in genetic technologies have put us on the verge of a new age of biology, we need to go into it with our eyes open. I think that's very sensible. The Economist back in 2018, talking about gene drives, said they promise great gains and great dangers. Don't ban and don't rush. And again, quite a sensible sort of position. And Faber et al., who wrote the recent paper on squirrels that I was talking about, well, on gene drives in squirrels, pointed out at the end of the paper that actually it would be very difficult to do the research at the moment. There's no legislative framework for the release of gene drive organisms. It's illegal to release grey squirrels, and once you have them in captivity, you must not release them. Indeed, if they get stuck in your garage, you mustn't release them because they're seen as such a pest. And there's no debate going on in Britain as to whether it would be acceptable or not, whether people would be happy with the technology, whether they would rather think of squirrels not being born, or whether they would be, rather squirrels disappeared. So the debate hasn't really begun. There's a lot of questions about whether people trust the science. The Royal Society has done a lot of work on this. Back in 2017, they tried to assess public attitudes to genetic technologies. And, it's a big report and lots in it, but I took two messages away from it. One was that attitudes depend on what it's used for. People who were asked, and it was a sort of focus group type arrangement, people in discussion differentiated between different kinds of uses. So they were pretty open. 80%, more than 80% of them approved of the use of genetic technologies to treat incurable life-threatening conditions, such as muscular dystrophy. Less than a third of them thought that cosmetic changes to animals, to pets, for example, making fish fluorescent, they thought that wasn't the kind of use that was so acceptable, or fewer thought it was acceptable. So it depends what they're used for. And the Royal Society argues that we need to have a discussion sort of not about the technology or not, but about the context within which it's used. In other words, something where people the public is encouraged to weigh up the risks, the opportunities, the benefits, and so on. The second thing is that trust in scientists depends on who employs them. If you ask people, do you trust science? Do you trust scientists? Which scientists tend to ask that question. I've been asked it lots of times. And it depends. They trust some more than others. They don't trust the general public in Britain doesn't trust scientists a huge amount. This is pre-COVID, mind you. This might well have shifted over the last 2 years. But 43% of people trusted university researchers, only 16% trusted scientists working within business organizations, or indeed funded by business, which includes a lot of people who work in universities. So that's interesting. And I think in terms for something like the Royal Society, thinking about science, the public is able to, in quite a sophisticated way, think about who they trust in terms of what the benefits are. And indeed, one of the conclusions about the GM controversy was not a distrust of at least initially, not a distrust of the technology, but a distrust of the fact that it was being trialed by a for-profit organization, and the social benefit wasn't very obvious. So, I mean, there are risks. I don't want to go into them in detail, but I mean, they're out there in the literature. Edited genomes will evolve. Edited species, organisms that have been edited in an unbounded ecosystem will move. They're freely mobile species. So fields and pens and greenhouses and islands are not biologically contained spaces. And therefore, it's important to recognize that possibility and to do the research on what's going to happen. So it's important that research is framed in a sort of ecosystem context. Genes move, we know that from GM crops. Engineered organisms get into new places. I put the rabbit up there because the rabbit is a confounded nuisance in countries like the UK and in Australia, but it's an endangered species in Spain. So one needs to be a bit careful about sort of the application of a genetic technique to a species which is a nuisance in one place and beneficial in another. And technologies can proliferate. I mean, if you'd done the genetic work to eradicate rats on a Pacific island, it would be that much easier to use the same technology to eradicate rats in the City of London or indeed in any other ecosystem. So there's a proliferation issue. There are things that need to be thought about. And therefore, there's a lot of interest within conservation starting to develop now about risk assessment and management. Goal assessment, this is work by Ronald Sandler of Northeastern University in the US. Is the goal of the project justified? Means assessment, can the goal of the project be accomplished responsibly? And desirability, is the overall project desirable? And the kinds of questions he's asking and wants conservationists to ask is, who has oversight of what is done? Who decides if a genetically engineered squirrel could be released into an Oxfordshire woodland? Who is it? The scientist? Is it the landowner? Is it the Nature Conservancy organization? Secondly, what's the role for the public? If this was your wood, the wood you walked in every day, would you have a role? And how do you deal with that? What do you do about enforcement? And what measures are there for the government to identify what has been done and then to impose it? It's something the courts can do. And what about jurisdictions? What about it's all very well to it's all very well to think that every country will make its own rules, but we're a very globalized ecosystem now, let alone everything else. So lots and lots of big questions. And this is just to say that it's not, for conservationists, it's not just them that are doing synthetic biology on wild living organisms. There's a lot of interest in forestry, in agricultural pest control, in public health. And so there will be, these are indeed the areas which will first see genetically engineered organisms developed using the new techniques released. And conservationists tend to say, well, we'll make our own decision about wild things. You can do what you like on your farms, but because those are domestic crops or industrial livestock systems, that's different. But we can talk about the wildlife, and I think that is an unrealistic barrier. So that's where we got to. Is it just another conservation technology? Well, it is a technology. It's not like the familiar ones. Conservationists cut, poison, burn, shoot, all kinds of things to achieve the outcomes they want. But this is new, the ability to engineer inside the genome. And I think it does therefore present for conservationists quite a big rethinking challenge to understand what these techniques can do, and to understand how they should be or should not be used. And in terms of technology, I rather like the comparison of the CRISPR and the chainsaw. Both the technologies both have biological and ecological impacts. The chainsaw does its work one tree at a time. It's piecemeal, it's slow, it's obvious to any observer. If you see, illegal logging is a problem, but it's at least conceivable that you can see it and fix it. CRISPR is different. It can reshape a whole genome. It's only observable by a scientist, and the effects are not confined to one individual, but can persist down the generations. And these differences explain, I think, why the technologies of gene editing are put in a different category in people's heads. They raise different concerns about risk and the willingness of society to accept them. So the last thing I think I'll say for conservation is that if you have spent 100 years protecting nature and saying it's valuable because it's natural, because it came by itself, because it's not human influenced, and then you start to use genetic techniques to engineer those species, then I think there's a real question about whether it disturbs this boundary between the natural and the artificial, and it makes the arguments of conservationists a little less convincing. And so I've talked to conservationists who are very enthusiastic about this, and I've talked to conservationists who are very worried about it, because it's basically, they say, it's domesticating the wild. We're going to be the ones in future who are making the decisions about how stuff should be, and that's not what nature is. And you say, okay, well, that's a very 19th century view of nature. And they say, yeah, okay, but that's what we do. That's what conservation is. And so these techniques actually undermine what we do. And I think that's the most challenging thing for conservation, is to think how. That's why we call the book Strange Natures. These natures are becoming strange because we can transform them as we can. How do we respond to that? How does conservation respond to that? I think that's the big question. Okay, that's me done. Thank you very much.
Thank you, Bill. That was fantastic. I'll just stop the recording and then we can get on to some questions.